Good afternoon, and welcome to Smithsonian Gardens webinar, Let's Talk Gardens. My name is Cindy Brown. I'm the Education and Collections Manager here at Smithsonian Gardens, and we welcome you. Thank you for attending this very special and different webinar. Today, we have Justin Kondrat with us. Justin is the orchid horticulturist, and he'll give you his fancy name in a second. But this webinar, since it's a tour of our orchid collection, we pre-recorded part of it. But Justin's here live to be able to start us off and to be able to answer any of our questions. As always, please put your question in the chat box and we will answer as many as we can at the end of our presentation. If you have questions that don't get answered, you can always send your questions to gardens@si.edu. Got to get the at for the mailbox, or to me, and I can forward them to Justin as well. So Justin, you have a lot to share with us. I'm going to leave, but I'll still be here just in case something happens. Good. And Zach is going to share your video when you tell him to do it.
Perfect.
And I'll see you at the end of the presentation. Take it from here.
Yes. Well, welcome everybody from probably all over the world, which is very exciting. I am Justin Kondrat, the lead horticulturalist within the Smithsonian Garden Orchid Collection. And this is the first time we've kind of offered this type of program to really see a behind the scenes look into what makes our orchid collection one of the top-notch living collections in North America and the world. And so with that, I will let Zach take it away. So enjoy the program, and I will be returning for any questions you may have. And please add any questions that you might have also in the chat, and we will try to get to as many as possible. Enjoy.
Hi, I'm Justin Kondrat, lead horticulturalist for the Smithsonian Garden Orchid Collection. Thank you so much for joining us today as I give a behind-the-scenes look at this amazing collection. This collection is very unique. It is almost celebrating 50 years since it began. This collection had many people involved, from staff at the Museum of Natural History, from horticulturalists, from passionate collectors, and over the years, from research expeditions collecting items for the archives, the collection grew and grew. And over the years, it has increased in numbers, been refined, increased in numbers, and been refined. Right now, we sit comfortably at roughly 4,000 collection items. With this space, we have a goal to increase our numbers to roughly 6,000. Collections, like Smithsonian Garden Orchid Collection, are a vital part of the Smithsonian institution. When many people think of the Smithsonian, they think of archives, documents, artifacts. But this is a great example of a living collection, similar to the zoo. This collection is a repository for germplasm of wild collected orchid species, and along with historic hybrids that have been donated over the years. The Smithsonian Gardens Orchid Collection is at a very unique spot, as utilizing the displayability of orchids, but also the conservation and research that comes along with this amazing flowering plant family. With over 30,000 naturally occurring species, Orchidaceae is one of the largest flowering plant families. Its uniqueness and diversity can be seen from the mountaintops all the way down to the mangroves and everywhere in between. We even have orchids in our own backyard in the Northeast. There are over 200 naturally occurring species in North America alone. So when people think of the tropics and orchids, they only think that they're found there, but that's far from the truth. Their evolutionary biology, symbiotic partnerships with mycorrhizal fungi, and unique strategies of adapting in various ecosystems have allowed for it to be one of the most successful flowering plant families known to everybody. A question that we often get here at Smithsonian Garden Orchid Collection is, how do you manage so many orchids? I have trouble just keeping 2 alive on my windowsill. Well, thankfully, there again, with engineers and years of dedicated research and troubleshooting how to cultivate many of these plants, we have this wonderful facility that we have here that allows us to cultivate so much diversity within this plant family. From humidity systems that allow us to keep proper humidity, to shade systems that allow for the proper lighting, from fertilizer injective systems, keeping our plants well fed, and heating systems and cooling systems, providing the correct conditions. We are able to culture many, many species and hybrids within this collection. The collection that we are touring today is comprised of only 15,000 square foot that is dedicated to the Smithsonian Garden Orchid Collection. So come along with me and enjoy the tour. One of the most important things when managing a collection of this size is ensuring that the plants are protected from various pathogens, whether we're talking about insects, fungi, bacteria, or viruses. It's making sure that we're safeguarding each collection item. The way I view this collection is that we're sort of in a hospital, if you will. It's kind of like it could be a petri dish of various illnesses or sicknesses. So it's making sure while the collection items are in this collection that they're cared for in an appropriate manner to make sure that they go on for generations. And one of those things is integrated pest management, so also known as IPM. So pretty much every single day when we're managing this collection, we make sure that we're managing these plants in a way that protects them from various pathogens. And this is done by using various tools and techniques to ensure the safe guarding of this collection. For instance, when it comes to handling the plants, we never touch a plant if we're grooming, repotting, moving a collection item. We always have compostable gloves on. And in between each plant, we always change those gloves to ensure that we're not cross-contaminating any collection item. For example, if I'm grooming this collection item, I have to change gloves before I go on to the next collection item. So if I'm making a cut, if I'm grooming, removing a flower, cutting into the plant tissue, that is really critical to change gloves. It also is important to use stainless and clean implements, whether you're cutting, dividing, grooming a plant. So here, our system that we've devised is a green bucket that has stainless steel scissors that you'd find in a hospital. These scissors are used once they're cleaned and sterilized. So the process is, this is in the green bucket, means it's good to go. I go and I work on a collection item. After I'm done with that collection item use, this is dirty now, it's contaminated. I put that in the red bin for the next use, and what will happen, this will go to a washing station where it will be use a detergent to clean and then followed by a sterilant. Then it will be rinsed again and then put into an autoclave where it is heated up and also pressurized to ensure that any pathogens that were on that tool are no longer viable. So always ensuring to have gloves on while working within the collection. We also have plates that we utilize. So this is just a regular catering plate that we utilize if we're going to be moving items. So there again, if I'm going to be shifting a plant to another bench or another area, or we're utilizing it for grooming or exhibition or photography, we place the plant on a sterilized plate, and then we can move that plant anywhere we need to, regardless if it's a porous or a non-porous surface. This allows us also to really make sure that we're putting a plant on a clean surface. Another way that I look at collection management is that every surface is contaminated. We don't know what is on that particular surface, from a fungus, from a bacteria, to a virus. So it's really critical to ensure that each collection item is theoretically kept in its own little bubble as best as we possibly can while we're managing this collection. Because there again, we are trying to preserve each collection item for generations to come, for future inspiration, scientific research, or just pure enjoyment. What do I have here? This is actually plastic moss that we utilize, that we're allowed to, we can sterilize and reuse when we're putting plants on exhibition or display. This allows us to ensure that anything that's touching the plant has been fully sterilized. Part of the system there again, when it comes to sterilizing and keeping surfaces clean, is using a detergent, followed after a rinse. Next is step 2, which is our sterilizer. So we use these in tandem to ensure that various surfaces are clean and free of also debris, but also possible pathogens that could infect our collection items. A powerful tool also is just your simple scouting. So there again, every week, usually on a Monday, I will go through the entire collection and I will put detailed notes if I found any particular pathogens or pest issues, where the plant is, if it's been treated, and how we are going to treat it moving forward. This allows us to keep our thresholds low, so the amount of pests that are possibly in the collection, so then we can manage each collection item and possible challenge that we have to the plant safety appropriately. And this allows us to look at various spikes in our collection, knowing that in spring we'll have various increase of other thrips or other insects that we deal with. So it's a great tool there again, mapping how many insects or pathogens are present in the collection at any given time, and there again being proactive and ensuring no damage to the collection is done. So part of our integrated pest management is reducing the amount of synthetic chemicals that we use on our collection items. And to do so, we enlist the help of beneficial organisms. So for example, we have particular mites that we release every 2 weeks. These mites actually are predatory on various pests that we're trying to control within our collection. The number one enemy is the thrip. The greenhouse thrip loves orchid flowers, just as our visitors and I enjoy. The one downside is to them is that they will damage the flower, discolorating the flower, but also they are a pathogen and a way to spread virus throughout the collection. So there again, ensuring that thrips are mitigated is critical to keeping our collection items safe. Another tool we use for IPM is when you walk through our collections, you'll see various sticky cards. So if you were to touch these are very sticky. And what this is if an insect sees this sticky card, they are yellow or blue for a particular reason, and the insect is attracted to this particular card, and when it goes, it gets stuck. And this allows us to scout to see if there's any pests that are in this collection. And it also allows us to track the insects as well. So it's a very, very helpful tool when trying to determine what the numbers are within the collection. Another part of communication when it comes within the collection is the utilization of these mini traffic cones. So these mini traffic cones allow us to communicate that this particular area once had a collection item, whether it's being used for exhibition or has been relocated, that this area has not been sterilized yet. So when we need to sterilize, we will remove the cone and then sterilize that area. So then we ensure that every inch of the bench has been cleaned, and if a new collection item has to be put back, or if we have to do some shifting, that can happen in a safe manner. The best horticulture practices also detail proper nutrition for the collection items. This allows the plants to actually be healthy enough to have their own defenses. So similar to you or I, we have immune systems. Plants also have immune systems. To help against the fight against pathogens, it's imperative for the collection items to receive the proper nutrients and care. This allows for those plants to fight off any bacteria, fungus, or viruses that are trying to compromise the plant's health. As you can see here, we really pay attention to details when it comes to ensuring that plants are a nice rich green color to ensure that they're receiving proper nutrients. We also, every 6 months, we send out leaf samples to get analysis done. This ensures that we're giving the plants the proper nutrients and they're able to absorb the nutrients. Another critical part is the physical capacity of each growing space and ensuring that collection items aren't on top of each other. There again, our goal is to provide the best growing conditions possible for each collection item. And this is done by allowing proper spacing of the collection items, ensuring that they're not growing into each other or shading each other out. So one of the imperative things that we do every 6 months is go bench by bench to ensure that all the collection items, if they're growing, most of them are growing pretty fast, you're giving them what they need, so they're gonna need to expand. And so that allows us, we shift various collection items within the collection to provide the proper growing conditions. And this also allows us, if the plants are healthy and getting the proper air circulation, this will also cut down on possible pathogens, such as with fungus and bacteria. One of the big questions I get is how do you keep all the plants healthy and growing? One of the main things that we do here at the gardens is ensuring that all the plants are repotted on time. So we have a schedule that we develop each year that outlines what collection items need to be repotted. There again, the repotting interims vary depending on what plant we're dealing with. In general, we usually do every 2 years, but there again, some plants are very aggressive growers, as we call them, and they like to walk out of their pots and kind of grow out of their pots. For example, put on my glove. This is a Cattleya hybrid, so the Cattleya Alliance. This is about 2 to 3 weeks out from flowering, so it kind of gives you an idea. This plant, of course, we wouldn't repot it if it's getting ready to flower. We would repot after it blooms. But just to show as an example, this is a plant that is definitely trying to venture out into the world. You can see the roots that are there again growing. This is an orchid that would typically be growing as an epiphyte, meaning growing on another surface. It requires a lot of porous space around the roots. This plant was potted about, I would say by the judge of it, probably 2.5 years ago. So we're about a half a year behind schedule with this one, but it happens. So the way we repot this Cattleya is, you see these are the old leaves and the old pseudobulbs. So those storage units allow for the plant to live in various areas. But you can see you have your old pseudobulbs and leaves where this one flowered, this was a flower, and this is soon to be a flower. So you see the older going to the newer. So there again, similar to a lot of other plants like an iris, how they kind of grow and they run, if you will. This will run away from where you put it. So we just have to catch them up. We're a lot faster than them, so we can catch up to them. So that's an example of that particular plant. What I would do is I would count back, rule of 2 to 3 pseudobulbs. So 1 pseudobulb, 2 pseudobulbs, and 3. And I would cut at the 3 And what I would do is place that old pseudobulb back against the terracotta pot so then it has room to grow into its new home. So as I said about porous media, we use a bark media here at the gardens for most of the collection items. This is an example of a bark that we use, and you can see it's pretty porous. We usually add other non-degrading materials such as charcoal and growing stone, but this just is an example of how coarse the media is that we use. There again, our repotting area, this is where a lot of transmission of virus could possibly happen. So there again, good horticulture is clean horticulture. So ensuring that the space is clean. Between each item, we sterilize this bench with a detergent, or wash it with a detergent and then sterilize following. And then we place, just to be careful, is we place another sheet of deli paper on this, for this particular collection item, just to ensure. And this allows us to place stuff, and it allows us to clean up our mess pretty easy. So what I would do with this plant, if I was to divide it, I would take this saw that we're able to sterilize. So this is just a regular sheath spathe that has the bladed end. And it's I am a pretty aggressive repotter. So there again, I would cut to the third pseudobulb and then only keep a few of the growths. And then the rest I would compost. Or if it is of conservation value, whether it's a historic hybrid or on the red list or critically endangered, I would keep the back bulbs and possibly share those with other institutions. And so that's part of how we would decide that. This plant, I would keep it in the same pot. And this is an example. This is all the hard work that pays off after it's been repotted. You can see reducing the pseudobulbs, and then you get the new growths that comes out, and this beautiful flower. This already had 3 sets of flowers before this. So it had more buds on this side and the other side that just faded. So this is, you're seeing this the last, it's probably gonna last another week and a half. If you could smell it, it's a beautiful floral fragrance. I just love that yellow throat with the beautiful flat white petals. And this is just a stunning orchid. This is actually named, there again on theme with our orchid exhibition. This is Cattleya Nancy Off, Linwood. So this is named after a very prominent orchid hybridizers, a very prominent woman in their family. To keep our collection safe from various pathogens such as viruses, we have a pretty extensive virus mitigation program here at Smithsonian Gardens. There again, with the scissors that we use for grooming and clipping that are used on collection items, that is also one of the major points where virus infection can happen from collection item to collection item. To mitigate that, we have developed a several-step system that allows us to help mitigate that risk. You can see here we have stainless steel scissors that were used on one collection item. After they were used, they were dropped into a red bin. That red bin was then brought to our sink where it was washed with a detergent, then rinsed, and then finally sprayed with a sterilizer. Let that soak for 5 minutes, and then it was rinsed again, brought into this bucket, the orchid wash bin. So this is ready, but they're still dirty. They haven't gone through their final stage. The next stage is loading them into this autoclave, which heats up. It's like pretty much a scientific high-end pressure cooker, if you will. Heats it up to, I believe, over 200 degrees and pressurizes at a high percentage. We put the items in here that we need to sterilize. We place those in there, and then we close. And then there again, we have to screw it on to make sure that it's pretty tight, because there again, this is very pressurized. We turn it on, and then there's a cycle that we use to make sure, so it's for the scissors. We run that one cycle, and then once that's done, we let them air dry. And then we place them into a green bin, which has been sterilized, and these items are ready to use on another collection item. One of the systems that we have here within the collection is to maintain proper humidity, as you can see. Most orchids that we cultivate here like a minimum of 55 degrees humidity. This means the amount of water vapor that is dissolved in the air around you. We also increase the humidity. Some plants like it around 75% humidity. And so wetting the floors would not do it justice. So in order, we have a pressurized water system that pumps small droplets into the air that create this fog that you see right now. And it is lovely every morning walking in when I pump the humidity up to around 65% humidity. And it's a lovely place to be. With so many collection items requiring various needs, we have to implement many indicators to communicate within this collection. For example, this is a reflector system that we have devised that allows us to find particular items that are critical to our collection. So in case of an emergency or items have to be evacuated, this is an item that has to be deemed appropriate and priority one, meaning that it has to be loaded into a truck or other procedures have to be put in place to ensure that this plant is safeguarded. So these reflectors note that. We also have pink flags that are around the collection. This notes that this plant needs a photo. So it needs either a floral shot or a high-res floral image. And then you'll also notice these large orange flags. So this is part of our IPM program. This allows us to note plants that we have concerns with, whether there's a pathogen that was found on this plant or something is going on that we have to take a closer look. So these flags allow us to easily locate these plants for scouting, but also is a visual cue to ensure that if there's a lot of orange flags, we need to do something. So this is a pretty cool plant. This is the first time in Smithsonian history that Smithsonian Gardens has named an orchid after this amazing institution. There again, this plant is Smithsonian Sunburst 175. So this is, you can see it's producing some flowers here. In about 2 weeks it will flower. This plant will then go on display for our visitors to see. This is a really exciting year. They're again celebrating Smithsonian's birthday with an orchid twist. So a really fun part of my job, of course there's a lot of fun parts, is building collaborations with other botanical institutions. This is a great example of a conservation collaboration that we formed with the Chicago Botanic Garden. This is a plant that was almost extinct in the wild. It is also known as the ghost orchid. It just kind of looks like a pile of roots. But if you looked very carefully, you'd see some scale-like leaves, but you have to have a magnifying glass to see them. But for all intents and purposes, this is a leafless orchid. It's named, there again, ghost orchid because it hangs on the trees, grasping, and all you see are the roots until it flowers. And when it flowers, it lets down this beautiful, pure white flower that dangles in the wind and usually blooms during a full moon cycle, so it reflects the moonlight. And it is fragrant during the evening and is pollinated by a night-dwelling moth that has a long proboscis to drink the nectar. This is a plant that has recently, there again, we've learned more about it so we can actually cultivate it under glass. This plant came from a wild collecting when it came to conservation programs through the Fish and Wildlife, working with some researchers at the University of Florida and the Chicago Botanic Garden. And we are very excited to have this in our collection now. Last year, it had a total of 8 flowers, which was quite magnificent, because I've actually never grown it before, so it was quite a horticultural feat for myself and for the team. A part of this collection is also making it accessible for not only the visitors that physically visit us at our museums, but also online. A part of our goals for the last few years for collection documentation has been getting all our collection items digitalized. What does that mean? That means to get all the images of both the flower and the entire plant shot available online that corresponds to our collection records. This allows for people from all over the world to view our living collections without even leaving their home. It is also a valuable resource for individuals trying to ID the plants that they have in their collections, or to see what we have. Another part of our facilities that helps keep our plants cool in the hot DC summers are these evaporate coolers. There again, we are in the cooler house, so this is almost running 24/7 during the summer months. But how this works is water is pumped up through these membranes, and you can see the water dripping through. This water, and as the air, there's fans that pull the air on the other end of the greenhouse that pull the air through these cooling membranes that allow for cooler air for air to be cooled by the water, thus allowing for evaporate cooling and evaporate cooling effect. One of the major tools that has been a game changer for the cultivation of our orchid collection is our rainwater harvesting system. So this has been the ability to harvest rainwater, collect it in a cistern, and then in turn water our orchids with that water. In the cultivation of orchids, pure water is critical. The ability to get rainwater has helped us tremendously. Before, we were actually on city water that contains a lot of various chemicals that negatively impact the culture of orchids. While using the rainwater has a low dissolved salt content, which allows us to understand what is exactly in the water and then add exactly what we would like for the water to have. This system here was installed a few years ago, and it's pretty much a mini computer. You can see there it has a screen that has our 10,000 gallon cistern. Right now we're at 3/4 full. This is a total of 8,141 gallons that we have to use for the orchid collection. Every time it rains, we have 4 gutters that are connected to the greenhouses, that there's a big pipe up on the rafters that channels the water all the way down this big pipe and then eventually outside to an outdoor cistern where the water is harvested. Once the water is in the tank, it is then pumped up using this computer to a desired PSI, and then it gives us our temperature. So right now we're looking at a chilly 53 degrees Fahrenheit. Pressure, we have micron filters that it goes through to ensure that we don't have any particles that would clog our lines, to make sure that we're not putting any debris within the collection. And then to, there again, make sure that we're not having any contaminants such as bacterial, fungi, or other various things. We have a UV sterilizing chamber that the water passes through and denatures anything that's going through it. Comes through, and then it has a pH right here. So our pH right now is 6.29. And so with orchids, typical pH, you want it to be 6 to 6.5. And so this is great if we're using the pure rainwater, but then when we use our fertilizers, they're acidic. And so that actually decreases the pH. So when we're watering, we actually have to add another buffer. So we have 2 injectors that work simultaneously to make sure when the pH goes down, there's a buffer that increases it to between 6 and 6.5 to ensure the orchids are getting what they need. That is to do with the availability of nutrients at various pH levels, so ensuring that we're giving them the optimal range so that they can absorb the nutrients that is in the water, but then also in the growing media that they're in. There again, orchids like tepid water. I mean, I know I don't like cold water, so orchids, why would they be any different? And so we have a water heater that helps temper the water. So we're on an average, the water that comes out of the spigot is roughly 73 degrees Fahrenheit. And that provides a good temperature to provide for nutrient uptake and also not to shock the orchid roots, which is really critical, especially when we're dealing with cold climates and seeing the water that is coming out of there at around 55 degrees, so that's pretty chilly. So ensuring that there's no shock that happens. And with this system, it's been critical in cultivating many of our orchid species that we really were never able to fully cultivate to success. And we've noticed a drastic improvement in the collection when it comes to flower and overall robustness of the collection items. So it has been a turning point and really providing the best environment for this collection. So we are at one of the rooms that many people don't get to see, really behind the scenes, besides being in the greenhouses. This is our inside of our facility of our boiler room. So this system that we have right here is our filtration system for our reverse osmosis system that helps feed our pressurized misting system. So there again, the reason we have reverse osmosis is it removes all the chemicals and various salts that are in the city water. And we end up getting a pure water that is right here, that is free of any salts that would lead to scaling or clogging of the various mist nozzles. There again, the mist that is being pumped into our greenhouses is at a very small molecule. So there again, any condensation or any particles would clog that, and there again, making the system not functional. So all the water gets filtered here and then goes to this 500-gallon tank and then gets pumped up over here through all the pipes to a pump that's right here that helps pressurize to 1,000 PSI. That water is then pumped out to our greenhouses where it feeds the mist nozzles to reach the desired humidity levels for the collection. So a part of this exhibition also allows us to get the plants on display. As you can imagine, a lot of these plants spend most of the year here at our greenhouse facility when they're waiting for the particular moment for them to bloom and come to the public. Also in preparation, I've compiled these collections items based on when they will bloom and also monitoring the temperatures that are in this greenhouse. So I've actually lowered the greenhouse temperatures to help preserve the blooms longer. So then when they go into display for the maximum freshness and longevity while on display. This plant here will be utilized during the orchid exhibition. So one of the criteria is that it's fully open, ready for display, fully groomed, but it also details that we have to get a printed label for the plant. So there again, so the public will know what they're looking at. So in order, we print these labels, there again, with where they're native to, what family, of course, Orchidaceae, and then Laelia anceps is this particular plant's name. This label will get festooned to a stake and will follow this plant during exhibition. So when you go to the Kogod courtyard, you'll see the plant and then its proper label. One of the challenges being an off-site facility 15 minutes from downtown Washington, D.C. is transporting these tropical collection items. A lot of these plants cannot handle temperatures below 55 degrees. And with their delicate blossoms, they can easily fade or be shocked. So in order to do this, we have to load all our collection items in these particular carts, and we have had custom covers that we've created for these carts to ensure that the plants are insulated. The plants will be loaded on a cart like this, all ready to go with the session labels and display labels attached. They will then be wrapped using this cover and then loaded onto our box truck, which is heated, and then brought downtown. And then eventually unloaded at a loading dock within one of the museums, and then eventually unrolled into the courtyard where they will be displayed, and then those plants will be placed in the appropriate beds. One of the most fun parts of my job is working together and doing the selecting of the collection items that go on display. So given this year's theme, I look at every collection item and look how it may attach to that particular bed and on display. Each collection item is selected based on how it relates to the theme of the exhibition and also where in that particular exhibition the plant could possibly fit. So for instance, if we're talking about hybrids that have been influenced and named after icons, iconic women, that makes me think of the First Lady's Orchid Collection that is housed at Smithsonian Gardens. And so the plants from that collection would go on display in that particular bed. While if we're talking about orchid collecting, we would gear towards more of the species, so the naturally occurring plants that would be placed in that particular bed. Or if a particular collection item has direct ties to the panels, so if there's a plant that we talk directly about, for instance, Darwin's orchid and Grecum's squescupedale, and we're talking about pollination relationships, that plant would end up in that particular bed. For the most part, we try to utilize as many collection items as possible because we know that most people don't get the chance to see these plants in person. There again, a part of the exhibition is also staging the plant material. So this is an example of our cymbidiums, just a few. We have over 150 within the collection. These are also known as the boat orchid. These are foundational plants that we use for our orchid exhibition. They are kind of the powerhouse when it comes to filling space within our exhibition. You can see a few, there again, staking of the cymbidiums. You'll see large bamboo cane stakes. Those have been done over the last few months, and they've been tracking the inflorescences or flower spikes as they've been developing. This is our cooler house, so a seasonally cool house. It gets down into the 50s at night and doesn't go above 60 during the day. The reason for that is to prolong these blooms and to make sure that they last as long as possible for the orchid exhibition. There are two plants here that are pretty cool. This is a Cymbidium species that actually was imported from China. So this is a wonderful species. It's just starting to open. So just in time for the orchid exhibition, it will be in full bloom. I couldn't have planned it better, but no, it just happened that way. The power of horticulture. We also have this beautiful plant here. This is a wonderful hybrid Cymbidium that we use for exhibition. So you can imagine these flowers, bringing them in the orchid exhibition nice and fresh. They will last up to 2 months in the orchid exhibition, so which is really quite wonderful. This allows us to bring them up, to take care of them properly up there, and then eventually to bring them back. And most of these cymbidiums have several spikes at various stages. So you'll have the ones that are fully about 20% open, and then you have others that haven't even started to break yet. So this allows us to really push the envelope when it comes to utilizing these particular collection items and to ensure that they're fresh as possible for our visitors to experience them. One of the great parts about a living collection is making sure that we are documenting everything correctly. From detailing if the plant has flowered, if it's been fertilized, if it's been groomed or repotted, or if something else has gone on with it. We utilize a wonderful scanning system to ensure that our data is uploaded correctly. Each collection item has a very, very securely fastened label that has the name of the plant, the accession number, which is similar to a social security number that you and I have. This allows us to track that particular collection item through the history of while it's in our collection. And then it also has a very nice barcode that allows us to utilize a scanner to upload our inputs or anything that's happening with this collection item. I simply create a task or note what's going on with the collection item, and then I scan. And when I scan, that eventually goes up to the cloud and is added to our database managed system. Also part of this tag is ensuring and doing phenology. So there again, you see this beautiful Laelia anceps is blooming right now. I would then again create a task system to ensure that we capture this, that it's blooming right now. I would scan, and then I use this little clip to clip it on the tag to ensure that we noted that we already scanned that for that particular week. We go through and scan the whole collection. Anything that doesn't have a purple clip, we scan so then we can track it. So for that month, this particular collection item has bloomed. This allows us to pull data at once to determine if there's times in the year that we have to increase our collection holdings of items that flower during that period, or if we have too many. Because there again, with a living collection that goes on display, our goal is to have a plant that is displayable 365 days of the year. So a question I often get asked is, what is your favorite orchid? Well, it's kind of like asking who's your favorite child when you have multiple children, or your favorite pet when you have multiple pets. I am partial to Pathogen, which is the Asian lady slipper. We have a few right here, actually. These are some bulldog type hybrids that we have in our collection. I just find them so interesting and unique. Most people think that they're carnivorous, meaning that they feed off of insects, but it's actually not the case when you look at the flower with that pouch. The pouch is there again all about pollination syndrome. They're again about its pollinator and attracting and decepting. So there again, what happens with this particular orchid is the bee or wasp would typically go into the pouch, whether it's being attracted based on pheromones or likes the look of the flower. It would go in there. It would get kind of confused. There's actually downward facing hairs, and the way the lip is curving, it kind of throws it off its tracks. But in the back of the flower, it has a ladder-like structure that the insect is able to crawl out. And when it does that, it comes out and has to push against a flap. And when it does that, the pollinia, or the pollen grains of that orchid flower, attach to that insect's back. And then the insect exits with some pollen on its back. And then if the bee or insect is silly enough or going to look for another flower, it will do the same thing, but except leaving the pollen behind in that particular flower. There again, leaving for cross-pollination. Really, really cool. And that's just one example of the complex relationships that many orchids have with their pollinators, and they've co-evolved over the millions of years. So I am very, very passionate about orchids, but then also public horticulture. I grew up in upstate New York, and I've always been enthralled by horticulture and plants and the way people interact with plants. I remember ever since I was little, I would garden with my grandmother. I would create this little paradise with sunflowers and other annuals and perennials. And it was a place I really could feel accepted and really just prompt beauty and intrigue with not just myself, but people around me. And I just love sharing that. I was fortunate enough there again to live down the street from a world-renowned orchid grower. And at the age of 15, he took me under his wing and showed me what I needed to know, there again, just the surface about orchids. From that point on, I just fell deeper and deeper in love with orchids, but still having that real deep passion for horticulture and how people interact with plants. I then found out you could actually work at public gardens and public horticulture. I did my first trip at the New York City Botanic Garden, and that's when I knew I had to be in public horticulture, whether it was at a horticulture level, working with public programs, or eventually a high-up leadership role. So right now, I'm kind of in between. I'm in the horticulture role, but I also help with public programming at Smithsonian Gardens and various educational avenues and how we connect our collections to our visitors. I found out that you could study public horticulture and learn about plant sciences and agriculture. I first attended SUNY Morrisville in Morrisville, New York, where I got an associate's degree in horticulture production. I was enthralled there again to learn even more about the plant sciences, and that's when I transferred to Cornell University and later earned my Bachelor's of Plant Science. And during that time, I was able to travel everywhere, from Hawaii to California, to New York, to Pennsylvania, to Florida, to also abroad, learning more about public horticulture and all the avenues that public horticulture can provide. And eventually I had to get a job. So I was fortunate enough to land a job at the United States Botanic Garden, one of our neighbors down the road at the Smithsonian Institution, where I worked within their orchid collection for 3 years, working out with their orchid exhibition and their collections. Eventually, the job over here opened up, and it was a golden opportunity to really get back into Smithsonian and really to help this top-notch collection into how we envision, and really to cement itself for future generations to ensure that we are looking at each collection item, holding on to it, and sharing it with the public for countless generations to follow. Thank you so much for joining me and our team as we walk through and talk about our collection at Smithsonian Gardens. This orchid collection is vast and diverse, and I hope I inspired you. And you too can also grow orchids at home or wherever you may be. But if anything, please come back and visit us, enjoy our exhibitions, or even visit us online for a digital experience. Thank you so much, and keep growing.
Whoa, Justin, thank you. And really, you're muted. Come on, make sure that I might have to unmute you again. Hold on, because you took off. There you go. Did it work?
There we go, technology.
Okay, good. The trials and tribulations of Zoom.
Yeah.
Thank you, everyone, for attending, and thank you, Justin, for giving such a thorough look behind the scenes of our orchid collection. And so people know we do have many of our orchids photographed and put on our website in a delightful program called Plant Explorer. So you can see many of them, their shining faces on those photos. Also, you can see them in our orchid exhibit, which opens in 2 days.
Yes.
Now, I know people are from all over the world, so you're not going to be able to come to our exhibit, which opens on January 29th in the Kogod Courtyard within the Donald Reynolds Center. And I always get the middle, there's one other word that's in there, but it's between the Smithsonian Art Museum, American Art Museum, and the National Portrait Gallery. So you'll be able to see all the pretty faces. And for those of you just wanted to see the pretty orchids and not see all the things behind the scene, we are creating a video of the exhibit, which will be just of the orchids in the exhibit, which include our collection pieces as well as some others that we have brought in for interest in, because they bloom for a longer time period. So you want to tell us a little bit about the orchid exhibit? Just the highlights maybe, Justin, because you're part of the committee that we all work together.
Yes, so it's Orchids, the Untold Stories of Groundbreaking Women. And so they're again focused on all the contributions that women have made and really the untold stories of a lot of these women that they've faced in science and various kind of old boys clubs, if you will. And so really focusing on also Smithsonian scientists that have contributed to our living collections and our conservation programs, whether it's at Smithsonian Gardens or at the Smithsonian Environmental Research Center, which is the North American National Orchid Conservation Center, which is housed there with most of our native orchid species. We, of course, right now we focus mostly on tropical species and hybrids. This exhibition will be, of course, in the Kogod Courtyard, which is a breathtaking museum. They're again wedged right in between the two museums, and our collection items will be on display there again, and it's going to be pretty fabulous. There's going to be about 300 collection items on display at time that will be rotated depending on how strong the blooms are, if their conditions are right, all that good stuff. We also do collaborate with the United States Botanic Garden. This is a partnership that we've actually been working together for over 26 years. Can you imagine that? And that together, we comprise one of the largest orchid collections in the world when we put all our numbers together. But there again, it's not all about numbers, it's about the quality of the collection items. And when those plants are on display, it's really quite striking, seeing both of our collection items and fully interpretive panels. And it definitely is quite wonderful. And there again, in this hybrid environment, we'll be having a lot of online programs, social media. So definitely keep plugged into Smithsonian Gardens, whether you're following on Instagram, Twitter, Facebook, or all the other avenues that we have, and also our website, which will be rolling out soon. I don't know if you wanted to talk a little bit about that, Cindy.
Well, first they want to know when the exhibit, how long the exhibit is open. So on the 29th, on Saturday it opens, and it'll be open until April 24th. Yes, it is open to the public. Everyone is invited. We do have COVID policies put into place for the safety of our visitors as well as the safety of our staff. And so if you go on to the Smithsonian Institution webpage, it will tell you what the procedures are at this point, because we're updating every day as circumstances change. And so I don't want to give you information that will be outdated tomorrow. So please go on and please look and see what the COVID protocols are in place right now. So that was one of them. Okay, now we'll extend a little bit for question and answers, but not much. We get off at 1. Justin's got to get back to taking care of those orchids. Somebody asked how many people take care of these orchids. Go ahead, Justin. You tell them.
So for our orchid collection, so it's myself as the lead of the collection, the helping steward the collection. But there again, we're all collection stewards of the collection. So myself, and then we have another full-time staff member, and then we have about 6 volunteers that help us with all the protocols and cleaning and just keeping the collection rolling. We also do have an educational program where we usually have at least 2 interns a year. So whether they're helping with installation of our exhibition or helping in gardening experience in horticulture within our collections. And then we do have, mostly that's about for the horticulture side, but then we also do have a plant records manager, Sarah Hadeen, who helps with all they're again behind the scenes within the computer realm of the mass sterilization, also with all our operating systems and various things. So that's that kind of side, but it's not too many people. You'd think it'd be more, but there again, we try to run a pretty tight ship here.
Yes, we do. But somebody also asked, do we have to decontaminate you and all the people that go into the orchid collection?
Fortunately, not yet.
No,
exactly. We, they're again using the barriers that we use, whether it's the plates or the gloves. That's fine enough when it comes to the protocols that we're dealing with and the viruses that we're trying to prevent. There again, there's very...
This is one of the reasons we recorded our video, because the internet connection at the greenhouse is really bad. So we were afraid it would freeze just like Justin is freezing right now. So looks like that, unless he pops in here, it looks like that may be the end of questions and answers this time. But please do email me. And my information is found on your webinar invitation, and you'll be able to send questions to me, and I'll be able to forward them to Justin so he'll be able to answer them. Unfortunately, we're not allowed to give you brand names of items that we use over the website, but contact me. We'll steer you in the right direction. But we're so appreciative of all of you coming to this outstanding presentation. We are really excited to show the behind the scenes stuff that you never get to see in the exhibit, and for all the care that we give to those very special orchids. I didn't get to ask Justin what the most rare orchid or the most valuable orchid is, and we do have several. Oh, here he comes back in. Maybe there he is. He came in on his phone. So Justin, one last question. What is the most rare or valuable orchid in the collection? And I'm thinking of Bucky, but I don't know if Bucky is the right one or not.
Yes, can you hear me? We lost power at the facility, so I'm on my iPhone. Never a dull moment. Thank you
Never a
for
dull
all
moment.
hanging in there. So, I mean, I would say, you know, we do have Bucky, or so it's Bucky as we refer them to, but it's Balbophyllum phalaenopsis Bucky. And that is they're again a large plant that is from Papua New Guinea that we have in our collection, and it has leaves that are up to 6 feet long that kind of look like a beaver tail. There again, we'll be sharing all these images. We'll have images with our orchid exhibition, and Bucky may be making an appearance. It's the first time that we're actually going to be having them debut in our orchid exhibition. I would say that, but then we also had plants that were wild collected in the 80s when they did collecting expeditions with the Museum of Natural History, which is, you know, there again, those are pretty cool because it has all the collection data and who collected them, the pressed specimens in the National Herbarium. So there again, it's all about the story that you can tell with the collection items. So, I mean, there again, it changes, but there again, as we have more of a conservation focus when it comes to the exhibition or our orchid collection, we're going to have a lot of rare, unique stuff that will be preserved for generations to come.
Thank you. And I'm going to wrap us up, but I'm going to say keep following us on our social media because we will be posting photos of those pretty faces in the exhibit as well as we do in the collection itself in the greenhouse. And please check out our website. We are going to have an expanded website for the orchid exhibition with videos, pictures, close-up looks at the interpretive panels, all kinds of things to be able to make your virtual exhibit or a virtual visit more exciting if you can't fly in from Kew or wherever else you are. So thank you again, and thank you, Justin. Terrific.
Yes, thank you. Keep gardening.
Okay. Bye-bye.
Bye-bye.